Tisdale. He's been my nutritional mentor. I first heard him speak 21 years ago in Chicago at a Glade camp. There was literally a group of, 12 of us there? 20? It was small, a few tables. Yeah, he went on for 3.5 hours, which he's not going to have today. But I first learned about insulin resistance and the importance of it. It really transformed my practice. So it was powerful. And then just more recently, he taught me about the importance of restricting protein. So you're in for a real treat. He's a very brilliant innovator, and he's just a wealth of knowledge about these molecular biological pathways. So welcome, Ron.
Welcome here. You've attended a lot of talks. Certainly a lot of talks that talked about the nutritional relationship between cancer and glucose, etc. How many people feel they have a pretty good handle now on cancer and the nutritional connection? Well, you know what's to come. My purpose today, I feel, will be actually to confuse you. I don't want you to think that you really know what's going on with cancer, and especially going on nutritionally with it. It's not so simple. But first, just a little bit of a background. And I've got a ton of slides because I don't want you to think I just dreamt all this up. So I'm going to be connecting the dots, but there will be a lot of dots. And I'm going to go through them really quickly. You're better off probably not to take notes. And what I want you to do is to try and get the bigger picture of all of this. And I will kind of summarize it for you right at the beginning. Now, Dr. Savory mentioned that around 2 billion years ago, there was what was called, actually, the Great Oxygenation Event. All of a sudden, basically, a bunch of photosynthesizing bacteria, cyanobacteria, put a bunch of oxygen into the atmosphere, and it was killing all life. Now, life had already been around for about 2 billion years. So for 2 billion years, which is half of life's lifetime, we were just single-cell organisms, bacteria basically, protobacteria, just floating around in the oceans and dividing. Life was very simple. But when oxygen came around, it complicated matters because it started killing everything. They weren't prepared for it. There weren't antioxidants. And so cells started to colonize and hide from the oxygen until some genius protobacteria decided that it would use the oxygen to burn food, and then we had a precursor to the mitochondria, and then, as was mentioned, these mitochondria were engulfed by other bacteria, and rather than being eaten, they became friends. And eukaryotic cells evolved. And there was a division of labor, a very important division of labor, when you started having multicellular organisms. Which cells are the most important, and which ones basically can be sacrificed to oxygen and other damage. We had the soma, the body, and the genome. The soma being the body, and then we had the genome. And it was decided that it was the genome that was important. It was the genome that nature wants to pass on to future generations. The body, the soma, is there to protect that genome from one generation to the next. Okay. So that division of labor has essentially dictated the rest of our evolution. And then cells had to decide, or I shouldn't even say cells anymore, multicellular organisms had to decide whether it was a good time to replicate that genome or whether those dwindling resources should be better served to maintain and repair the soma that's taking care of that genome so that it could replicate at a perhaps future more opportune time. And that critical decision, of course, is going to be dependent on nutrient availability. And that nutrient availability had to be sensed. So there were nutrient sensors that detected the nutrient availability that then allowed the cell to make that critical decision of whether it should replicate or whether it should harness the resources towards maintenance and repair. And so we have one important principle, and that is that, number one, maintenance and repair versus replication is a critical decision that all cells are making all the time, and that is really relevant to cancer. And number two, that our genes don't do anything. They just sit there. So this thing about genes causing cancer had to be ridiculous right from the beginning because they don't do anything. They listen to instructions. They're just books. It's genetic expression that dictates everything. And that genetic expression is going to be more dictated by these nutrient sensors than anything. In fact, I came across an article not too long ago that showed that just changing from a low-fat to a high-fat diet changed over 8,000 genes. Well, we have about 18,000, so almost half of our genes were changed. The expression of those genes were changed just by changing the macronutrient composition of the diet. Well, that's a lot, because the genetic expression between a man and a woman difference is about the same. In other words, you can change yourself as much as you would if you actually did change sexes just by changing what you ate for dinner. That's huge. That's how powerful these nutrient sensors are. Now, aging is a battle between damage and the repair of that damage. And the repair of that damage is controlled by nutrient sensors. In other words, aging, we know now, is actually controlled by nutrient sensors. And that's been shown now for probably 15 years. The changes that one sees when they do experiments on worms and flies and mice and all sorts of different species are virtually the same among all of them. These nutrient sensors are virtually unchanged since eukaryotic organisms, since nucleated single-cell organisms. We had nutrient sensors, and they are the same 2 billion years ago as now, and vice versa. So these are really critical to our life. Aging and cancer are also intimately connected. All these experiments done on calorie-restricted animals, or all these genetic experiments done on flies and worms, especially mice and other animals, not so much on the worms because their cells don't reproduce, but on mice, for instance, they die of cancer. So you're not going to extend life unless you diminish cancer. There's actually much more profound reasons why cancer and aging are so tied together. I can't go into that today. But that's just a trust you'll have to have. That cancer and aging are essentially married. If you reduce the rate of aging, you will reduce the rate of cancer. You treat cancer by actually treating aging. And if you don't treat the actual roots of aging, you're really not going to get to the roots of cancer either. Okay? So that's just kind of the background. And now we shall move on. Now I've got a whole bunch of slides. I'm going to go through them really quickly. And a lot of it I'm just going to be reading right off the slides. So you can see what some very well-known people had to say about some of these nutrient sensors. We're going to go through the major ones, insulin, leptin, and the TOR pathway, which Joe alluded to a little bit earlier, which is the most important one. But first, you can have the answer. So if you have to leave early, all you have to know is this: that your health and lifespan is going to be determined by the proportion of fat versus sugar you burn in a lifetime. That's it. If you burn fat, you're going to be healthy. If you burn sugar most of the time, you're not. And almost everybody is burning sugar most of the time. They're not going to be healthy. Whether you burn fat or sugar is going to be determined by nutrient sensors. And those nutrient sensors are controlled by what you eat. That's really all you have to know. But now if you want to know the details, here they come. This is from that talk. And Joe mentioned a talk in 1995 at GLACAM. And I actually found some of these slides. They didn't have PowerPoint at the time. They just had slides, with a little slide projector, and they'd always got stuck. But here I was talking about hyperinsulinemia and how it adversely affects all degenerative diseases. So 25 years ago, a quarter of a century ago, I was an insulin guy. I introduced insulin to the world pretty much and thought that it controlled the center of the universe. It controlled all chronic diseases. Now, people are starting to think that today. So here I was talking about its connection with coronary artery disease, hypertension, cancer, stroke, diabetes, obesity, autoimmune disorders, mental decline. Another slide talked about the intake of macronutrients and the risk of breast cancer. Again, this is January 1995. The risk of breast cancer decreased with increasing total fat intake. You eat more fat, you get less cancer. So this is not new. Okay. This is stuff that was known 20 years ago. It just takes that long for this stuff to get implemented, which is really unfortunate. The risk of cancer increased with increasing intake of available carbohydrates, starch being the chief contributor. From insulin and its metabolic effects, a few years later, insulin increases cellular proliferation. What does that do to cancer? It increases it. So we're still in the 1990s. Then I had a nice teleclinic with Joe in 2005. Two of the major factors that will determine whether a person has cancer are the amount of sugar and insulin. Sugar is just listening to orders. It's listening to orders from insulin and leptin. You get insulin and leptin right, your sugar is going to be right. However, the converse is not true. You can bring down sugar with drugs, but you're not really improving that person's health if it's causing insulin and leptin to increase. All you're doing is increasing your risk of cancer. In other words, you have to control the nutrient sensors. It's not just a matter of controlling sugar. Sugar is doing nothing but listening to orders. The problem is not with sugar. The problem is what sugar is listening to. For instance, diabetes is not a disease of blood sugar. It's a disease of nutrient signals. It's a disease of insulin, actually more importantly leptin, even more importantly mTOR, you'll see. But even if you just get to the point that it's a disease of insulin, you're ahead of your doctor. It's just treating diabetes by trying to lower the blood sugar with blinders on, mostly by raising insulin. So, from here on, this is what you have to know. That the most useful piece of learning for the uses of life is to unlearn what is untrue. Similarly, our ability to open the future will depend not on how well we learn, but on how well we are able to unlearn. Okay. And I had to do that. As I say, I thought insulin was the center of the universe. Even above glucose, I never really got to glucose being that important. It was insulin. I knew right from the beginning, a quarter of a century ago, that insulin and other things control blood sugar. That it wasn't going to be sugar that was going to be the answer. It was going to be the nutrient sensors. The first one I thought was insulin. Okay, now is where you have to unlearn. And I will apologize. Don't kill the messenger. I'm just going to be repeating what others are saying. Okay. Unraveling the role of glutamine in tumorigenesis. Increased metabolism of glutamine is now recognized as a key feature of the metabolic profile of cancer cells. We will discuss the roles of glutamine in contributing to the core metabolism of proliferating cells by supporting energy production and biosynthesis, and cells coordinate glucose and glutamine as nutrient sources. That's known. That was already mentioned. Glutamine is a prime fuel for cancer. But the important part there is that glutamine is metabolized in mitochondria. If you're going to use glutamine as a fuel, it's going to be metabolized in mitochondria. So they can't be too dead. We know this. Simple sugar lactate is like candy for cancer cells. Cancer cells accelerate aging and inflammation in the body to drive tumor growth. The researchers, lactate is like candy. Cancer cells are addicted to this supply of candy. Cancer cells love lactate. What do they do with that lactate? They burn it for fuel. Where? Well, new information. that it's burned in the mitochondria. They can't be too sick. Okay. We know that lactate is a fuel. What is it doing with it? It's not just the core recycle, and that's not just being recycled back to glucose for glycolysis. It's being metabolized in healthy mitochondria to be used as fuel. Ketone body utilization drives tumor growth and metastases. I know this created a lot of uproar. They didn't think this was a true study. But this group has studied a bunch of papers to this, and there's another one that just came out in Cell Research. Hepatocellular carcinoma redirects ketolysis, meaning metabolism of ketones as fuel, for progression under nutrient deprivation stress. So just like us. If you go into what the body perceives as nutrient deprivation, you use ketones. Well, so does cancer. Cancer loves ketones, as a matter of fact. So giving cancer ketones is not going to be your answer. Well, we got more. We're just starting here. Cancer also uses fatty acids. Okay. Researchers examined the use of glucose and glutamine by tumor cells. Many cancers instead prefer to metabolize fats. Despite the pervasiveness of this phenotype, knowledge of the pathways that drive fatty acid oxidation in cancer is limited. This just came out actually last week. But we've known actually for some time, they just didn't know how it happened. What this study was showing is actually how cancer is using fatty acids as fuel. And of course, fatty acids are only metabolized in mitochondria. Again, they cannot be that sick. And these are some important cancers, like prostate cancer, leukemia. They prefer fatty acids as fuel. Now, this is an important, and I could have put literally 500 studies here. My background is in the biology of aging. I've studied that for 30 years. And I will tell you right now that the free radical theory of aging is dead. Nobody talks about that anymore. There is an offshoot of that called the mitochondrial free radical theory of aging. And that's not dead yet, but it's very much dying. There's a lot of papers that have disproved it. This being one, we review findings that suggest that the age-dependent mitochondrial dysfunction is not sufficient to limit lifespan. Furthermore, mitochondrial reactive oxygen species are not always deleterious and can even stimulate pro-longevity pathways. In other words, they've shown that when you damage mitochondria up to a point, it elicits a response that actually extends lifespan. It's called hormesis. It's like what Nietzsche said, that which doesn't kill you makes you stronger. Well, that's especially true actually in aging. If you actually increase reactive oxygen species, increase oxidative damage in mitochondria, in many different species, up to a point, they live longer. It doesn't kill them. It doesn't cause cancer. The mutator mouse is what they did is they mutated a mouse so that it could not manufacture properly DNA polymerase, which is used to repair DNA. So these mice had a mutation burden 30 times higher than the controls, 30 times higher, and it showed no deleterious effects. Okay. And we're talking about mitochondrial oxidative damage. So this is mitochondrial DNA polymerase. So these mitochondria had 30 times the mutations of control cells and showed no ill effects or limited lifespan. And again, the studies that disprove the mitochondrial free radical theory of aging are numerous. I mean, you can just compare different species. Parrots live 100 years, and they have the same rate of oxidative damage as other birds that live 4 or 5 years. The naked mole rat is a kind of a popular species to study aging because it lives over 30 years, whereas comparable rats live 3 to 4 years. And it, by the way, suffers tons of oxidative damage. And all they're saying is somehow it just tolerates it. In other words, the oxidative damage is not what drives aging. In the naked mole rat, in the parrot, and likely in us. Does it cause damage? Yes. But aging itself is a balance between how much damage we acquire and how well we repair that damage. If we could repair damage as fast as it occurred, we would live forever. And if we go back to that nutrient sensor controlling the genetic expression of maintenance of the soma versus replication of the genome, we see that nutrient sensors are really what controls repair. So it's not the damage, it's how well the nutrient sensors are able to upregulate the genetic expression of repair that will cause the phenotype we see as aging, which means cancer. Growth hormone. Now I'm going to have to start going really quickly. I don't know what time I started. About 5:15? So I have about half an hour. Well, I had more than 15 minutes. Just because I started late. It wasn't my fault I started late. No, I started at least at 5:10. Okay, calorie restriction, slowing aging and extending life. Basically, what they have found in calorie-restricted animals is that the cause of calorie restriction, first they thought was insulin. Then it was shown it wasn't insulin, it was actually IGF. And you have to understand that back in our evolutionary history, insulin and IGF were one and the same hormone. Okay, they diverged much later in our evolution. And so most of the adverse effects that we attribute to insulin are actually attributable to IGF. Now, IGF is a growth hormone. Okay? And if you keep upregulating that growth hormone, IGF, you age faster. And you get cancer. And if the previous slide showed if they disrupt the growth hormone receptor, that animals live longer. And actually, a friend of mine did that, Andre Bartke. Wonderful guy who got the first Methuselah Prize for aging. It's a $1 million prize, the biggest prize in aging research, for allowing a mouse to live twice its normal age by disrupting IGF communication. The role of insulin and IGF in longevity. Insulin and IGF initiate actions via highly homologous signaling systems, which is what I just mentioned. And the common and consistent effects of calorie restriction in rodents has to do with lower circulating insulin and IGF concentrations, increasing insulin sensitivity, lowering body temperature, fat-free mass, and lowering thyroid. That insulin and IGF are a family of hormones that regulate metabolism, growth, cell differentiation, and survival. So we went from insulin early on in aging research, about 17, 18 years ago. Cynthia Kenyon and others implicated insulin. Then it was really more directed towards IGF. And I actually met Andre Bartke at a meeting about 20 years ago, and I was an insulin guy, and he was IGF. We became good friends because of that, because nobody believed us. But now it's very well accepted that the insulin and IGF pathway have a very instrumental role in aging research, but it's not the ultimate role, which we will see. And strong similarities exist between insulin and IGF signaling in worms, flies, mammals, humans. In other words, these nutrient sensors are virtually the same throughout all multicellular life. And we'll see even beyond that. Dwarfism. This is Laron syndrome. It's a family in Ecuador that has a genetic defect in their IGF signaling pathway, and they virtually do not get cancer or diabetes. In yeast, worms, and mice, restricting growth hormone makes these creatures live longer. This doctor from that area had diagnosed the family with Laron syndrome, a rare syndrome caused by a gene mutation. Over the course of his years with those family members, he noticed that these people were almost completely devoid of cancer and diabetes, an observation that squared with the research of Longo and others who had done research on yeast and other species to show that IGF plays a huge role in the aging process and cancer. It could be because these cells must invest energy in either trying to grow and reproduce or to protect the genome. In other words, food wasn't available indefinitely. There was always a problem with food availability, and cells have to decide what to do with it. And that major decision is, do we invest in maintenance and repair of the body, the soma, or do we invest the nutrient resources in cellular replication, in which case the body becomes irrelevant. This is another corollary then you have to understand, is that nature does not care about you living a long, healthy, post-reproductive life. Okay. What's natural is to replicate your genome, make babies, and then die. That's natural medicine. What we're practicing is not natural. I would like you to live a long post-reproductive life. You're not going to be able to do it by following Paleolithic nutrition, for instance. You're going to have to do it by following the science of aging, see how nature has allowed us to replicate, to live long enough to repair our soma and our body so that we can replicate and do her bidding, and then apply it post-reproductively. Ungrowth hormone increases longevity. They came up with something that inhibits growth hormone, and these people lived, or these animals lived longer. It's a growth hormone antagonist. So all these people taking growth hormone as a anti-aging therapy are in for a rather big surprise. It just won't last very long. Long-term effects of calorie or protein restriction. Okay. Our data provide evidence that protein intake is the key determinant of IGF. Okay, now we're getting to the nuts and bolts here. You're going to see this word protein a lot. Protein drives nutrient sensors more than glucose does. Not the least the reason is protein can be made into glucose. Glucose can't be made into protein. Insulin analogs and cancer risk. Insulin functions as a growth factor and therefore can proliferate tumors. Okay, that's well known. A little bit about leptin. Does everybody here know what leptin is by now? Nobody? Oh dear. Okay, we're going to have to go through this really quickly. I'll describe it really quick. Leptin is a hormone that is made by fat and other tissues. Its primary function is to tell your brain how much fat you have and whether you need more or less, because food wasn't available 24/7. So our ancestors always encountered some famine. And we had to store some fat, but we couldn't store too much, because if a lion was chasing a group of people, who's it going to catch? The fat one, they couldn't make it up the tree. Yeah. And of course, a fat person wouldn't be a very good hunter-gatherer either. So you wanted to store some fat to withstand a famine, but not too much fat that you're going to be dinner for a lion. So fat had to be controlled. And leptin is the biggest controller of fat metabolism. And it will signal, if you have too much fat, your fat produces more leptin, goes to your brain, the hypothalamus, and it tells your brain that you've got too much fat, you better burn some off and stop being hungry. Nature knows that the only way you're not going to eat is if you're not hungry. So it very powerfully prevents you from being hungry. If you stimulate that area of the hypothalamus, you will not eat. In fact, you will starve to death. They've done that in mice and other animals. Right adjacent to it is a hunger center. If you stimulate that, you will continually eat. You will be so hungry. Okay, that's leptin. But it does much more than that. It controls virtually all of the functions of the hypothalamus and regulates virtually everything that has to do with fat intake. We know that leptin, for instance, regulates gluconeogenesis centrally, through the vagus nerve. Leptin mediates inflammatory substances. So we know your fat makes a bunch of inflammation, inflammatory. It makes aromatase that converts testosterone into estrogen, which is why fat men have breasts. Leptin does a lot. It controls, to a large extent, the sympathetic nervous system, how fast your heart beats. It's a really important hormone. And so then I went from insulin to leptin, wrote a book about leptin and told about its importance, about 15 years ago or something. And it really is important. But it's found in only multicellular animals that store fat. It's the central nervous system control of food intake. And here it says central or pharmacological studies suggest a more critical role for leptin than insulin in mammalian energy homeostasis. And that is a true statement. Okay. Leptin controls insulin more than insulin controls leptin. Chronically elevated leptin decreases hypothalamic leptin receptor, and what they're talking about is leptin resistance. The more leptin you're exposed to, the more insulin you're exposed to, you get leptin and insulin resistance. Then there's a disconnect. That's how people get fat. So once your hypothalamus starts becoming resistant to leptin, you start getting fatter and fatter. You make more and more leptin, but your hypothalamus doesn't hear it anymore. It thinks you're skinny. So there's a disconnect. So it'll keep you hungry until you get fat enough that you produce more leptin so that it can finally scream to your hypothalamus that you've got enough, even though you've got 100 pounds too much. What you've got is failure to implement. Okay? You've got a failure to implement insulin signals, a failure to implement leptin signals, and what I've said for 20 years, and I've not found an example to contradict this, is that all chronic disease is due to miscommunication. That's what you have when you produce up. But leptin is signaled more by glucose and protein. It's not signaled by eating fat. So even though it's produced by fat, there's kind of two stages to leptin. Nobody seems to know this at all. Everybody, that is even studying leptin thinks that there's kind of a steady state of leptin that's dictated by how fat you are. That's not true at all. You can double your leptin in one day if you eat a high carbohydrate meal. A high carbohydrate, high protein meal, you're going to double your in that one day. And it's those peaks of leptin that I believe drives leptin resistance. And then you get what they call a vicious cycle of escalating metabolic devastation. Indeed. Leptin regulates pro-inflammatory immune responses. The link between high leptin and telomere shortening was even stronger than the link with obesity. Telomeres, a lot of people are attributing, as a cause for aging. It's really not, but it is important in cancer. And the reason that we have telomeres, I think, is because it limits the number of times cells can divide. People know what telomeres are. And they're like little, they call them like the ends of shoelaces, and a little bit is snipped off each time a cell divides, so you have about 60 cell divisions. If you have cells in culture, the problem is cells produce telomerase. All cancer produces telomerase, so they can lengthen the telomeres to whatever they need. But the problem is more the other direction. When your telomeres get short, it's very inflammatory. And what telomeres really are, and I wrote an essay on this a while back, it's basically just an expensive marker of inflammation. It's how rapidly your cells are turned over. They're especially measuring white blood cells. So when you have shorter telomeres, it just means that your white blood cells are turning over faster, and a lot of that is due to leptin. Proteins and hormones stored in body fat, such as leptin and insulin, and insulin-like growth factor, promote prostate tumor growth in obese men. Leptin's failure may be considered for the metabolic decline seen with aging. There are a lot of people who feel that leptin resistance is a major cause of aging itself because of how the broad ramifications. We know that leptin, I should mention, also controls reproductive behavior. When leptin was first discovered, they thought that they had found the holy grail of obesity. It was a mouse. And Jeffrey Friedman at Rockefeller University in 1994 figured out why this mouse was diabetic and obese, and he found it was deficient in leptin. And so he synthesized it, he injected leptin into this mouse, and in 6 weeks it became thin, non-diabetic, and very healthy. And the pharmaceutical companies thought that they had the answer to obesity, and they were all tripping over themselves to find to patent the first genetically induced leptin injection for people until they measured leptin in people, and they found out that it wasn't like these mice who had low leptin, that almost all fat people have too much leptin. So giving them more leptin wasn't going to help. They were leptin resistant. And we haven't been able to figure out how to treat insulin resistance, let alone leptin resistance. So all those drugs that they're calling insulin sensitizers are not at all. They're nuclear receptors. We'll get to that. Don't have time right now. Long life was predicted by low levels of leptin, meaning it's more sensitive. Okay? If you want to be able to smell something in a room, you want to keep the odor down. If you stay in there under a high odoriferous condition, pretty soon you're going to lose your smelling. You're going to lose your hearing if you're overexposed to noise. You want to keep it down. What does medicine do to treat diabetes? They increase insulin. And the cause of diabetes is due to too much insulin. And yet they just make it worse. Again, that's why the science is important. The regulation of leptin secretion is by, look at this, amino acids. Again, protein. But glucose does too. So glucose and proteins are what actually regulates the secretion of leptin from adipocytes. A new drug to tackle the fat problem. Now that they know the atomic structure of the receptor, they can design a drug. We don't need to design a drug. We just don't eat a bunch of carbs and a bunch of protein. It'll work better than any drug to keep leptin down. And I measured leptin in I've measured insulin and leptin in 1,000 people. Now I will promise you that if you eat a necessary protein and not excess protein and a very low carbohydrate, meaning you have one choice, is a high-fat diet, you will dramatically lower insulin and leptin, and you will lower leptin in 1 day. Insulin takes longer. No. No, you got to at least give me the time I was supposed to have. 45 minutes, not a half an hour. There's no way. Protein increases IGF-1. High protein accelerates aging. Reduced protein extends life. We get to TOR. Okay. Now, Joe mentioned TOR. The quick summary on TOR is that all of these other nutrient sensors feed into TOR. It's TOR that makes the decision. They don't work autonomously. Glucose does not work autonomously. Okay. TOR makes the decision whether cells should divide or not. That's well known. TOR is old. It's found in all eukaryotic cells. It's older than insulin. It's the oldest nutrient sensor we know. It's so old that it takes instructions directly from glucose and protein. It doesn't even have to have insulin or IGF. High glucose and even most importantly high protein will stimulate TOR. And it stimulates TOR to change the genetic expression away from repair and towards cellular reproduction. Here's what you have to know. All I want you to do is, if you can look at the top, I don't know if you can see it, but you will see amino acids at the top, you'll see growth factors, you'll see glucose. So way up on top there to the left, you'll see amino acids. All the way to the right top is glucose, and then growth factors are kind of in the middle. And they all feed into this intricate network of TOR, which actually consists of two complexes. The actual TOR molecule is very ancient. There have been other molecules that have built upon it, depending on the evolution of other nutrient sensors. Okay. But all nutrient sensors feed into TOR. There's no exception to that. And it makes the final decision whether cells should replicate or not. Glucose doesn't just act on its own. If you take a bunch of glucose, it doesn't just cause cell division. Okay. It goes into TOR first, and it reads all the signals, and then makes kind of the final decision. Do we have enough nutrients for cells to divide or not? You keep pushing TOR towards cell division, you get cancer. And I should mention that today we know that calorie restriction, the benefits you see from calorie restriction, including reduced cancer, extended lifespan, reduced diabetes, all the benefits you hear about with calorie restriction now is all attributed to TOR. There have been, over the last probably 10 years now, hundreds of articles of support for that. And there actually is not a whole lot of debate on that anymore. TOR is the major mediator of calorie restriction. TOR signaling and control of growth through the growth factors, through It says it was discovered in yeast, but is conserved in all eukaryotes, all cells that have nuclei. So we're going way back, 2 billion years is TOR. And it controls, it's the same in worms, flies, mammals, controls growth in response to nutrients, e.g. amino acids. Okay. Amino acids, I should mention, it's been shown quite definitively, is the primary signal for TOR, more than glucose. Glucose, insulin, IGF, all stimulate TOR. The most powerful signal for TOR is protein, and even specific amino acids in that. TOR is implicated in development and aging, many major diseases, including cancer, cardiovascular disease, inflammatory disease, metabolic disorders. We know it's implemented in diabetes. The TOR pathway is important in many types of cancer. That pathway activation is one of the most common molecular alterations in human cancer. Elevations in TOR are found in at least 70% of cancers. Another thing that's important is that TOR coordinates mitochondrial activity. So when you talk about the decline in mitochondria, you're talking about an actual increase in TOR, because then it reduces what's called mitophagy. Again, whether we age or not, whether we get cancer or not, is going to be determined by how well we repair damage. It's not whether mitochondria get damaged. That's actually irrelevant. What is a matter is whether we can get rid of that damage. And we get rid of that damage by a variety of mechanisms. The ultimate mechanism is we eat up the bad mitochondria. So there's a process called autophagy, which is regulated by TOR. When you keep TOR low, it's a signal there aren't a lot of nutrients available. Where is it going to get the nutrients it needs to live? From yourself. It will actually pick out the damaged proteins, damaged mitochondria, and digest them, and recycle the parts for energy and components to maintain the cell. Whether we get cancer or not is going to be determined by how well we repair damage. It's not whether mitochondria get damaged. That's actually irrelevant. What is a matter is whether we can get rid of that damage. And we get rid of that damage by a variety of mechanisms. The ultimate mechanism is we eat up the bad mitochondria. So there's a process called autophagy, which is regulated by TOR. When you keep TOR low, it's a signal there aren't a lot of nutrients available. Where is it going to get the nutrients it needs to live? From yourself. It will actually pick out the damaged proteins, damaged mitochondria, and digest them, and recycle the parts for energy and components to maintain the cell. And that's why they feel, and in fact, very powerfully feel, that almost, if you want to use the word proven, that that's a necessary and sufficient condition to extend life and get the health benefits of calorie restriction, is because you are increasing autophagy and mitophagy. TOR controls ketogenesis. If you're going to produce ketones, you're going to go through TOR. Amino acids mediate TOR. TOR is activated by free amino acids. Here's calories do not explain the life extension in so-called calorie restriction. Okay? It's not calories anymore. That's been shown quite definitively. It's what kind of calories, and basically it's those calories, the type of calories, mainly amino acids and glucose and nutrient factors, that control TOR. That's what controls calorie restriction. The ratio of macronutrients, not calorie intake dictates cardiometabolic health, aging, and longevity. The interesting part here is they compared carbohydrates with protein. And they showed that even if you increase carbohydrates, if you limit protein, you extend life. In other words, it's more important to restrict protein than carbohydrates. And again, when you're extending lifespan, you're talking about restricting cancer. It doesn't mean that you want to eat a high carbohydrate diet. Please do not get me wrong on that. Do not misquote me on that, because I didn't say that. Okay. I was one of the very first, there might have been 4 or 5 people in the world when I was recommending a very low carbohydrate diet, but I, right from the beginning, was probably the only one that started recommending a high fat diet, because I recognized the problems with protein right from the start 25 years ago. TOR wasn't even known at that time. This just supports it even further. I've not changed my dietary recommendations over the last quarter of a century, with the exception that now I will say we can probably get by with even less protein than I was recommending 25 years ago, and I wasn't recommending very much then. It says, longevity can be extended in ad libitum fed animals by manipulating the ratio of macronutrients to inhibit mTOR activation. Okay. That's critical. That's what you need to do. Your diet needs to be constructed to inhibit mTOR. The TOR pathway regulates gene expression that rapamycin, I didn't really mention what TOR stood for. TOR stands for target of rapamycin. I don't have a lot of time to tell you the history of TOR. It's very interesting. It was found in Rapa Nui, which is an island in a part of the Easter Islands. It was actually found in the 1970s. A pharmaceutical company sent a group of scientists to just pick up soil samples because it was a very remote location, and they were going to see if they could find anything in the soil with some kind of antibiotic, and they found an antifungal. It was very new. That was pretty cool. They called it the target of rapamycin. They named the drug rapamycin that they found, the antifungal. But it stood on a shelf, and they started doing experiments on it about 20 years later. And they said, whoa, this is very interesting. It's an immune suppressant. So they patented it as a immune suppressant. And they were giving it to kidney transplant patients. And they found out that even though it was depressing the immune system, and I won't go into exactly why it does that, the kidney transplant patients who were taking rapamycin that depressed TOR lived longer anyway and had a lower incidence of cancer. And then they started doing tests on animals, and they found out, yes, almost uniformly, they live longer and they have a reduced incidence of cancer. There's 2 TOR complexes. One mediates, they mediate different things. I won't go into the complexity of it all. But it was a very cool finding, and they're still doing research on that. The effects of dietary protein restriction on glucose and insulin. In other words, you restrict protein, you actually have diabetes as much as if you restrict carbohydrates. A low-protein diet extends lifespan in flies, the so-called anti-Atkins low-protein diet. Atkins was actually a high-protein diet, not a high-fat diet. It was later morphed into that by actually a friend of ours. It goes way back, who unfortunately passed away a couple years ago. This is a drug that they have to target mTOR, and they've shown that it reduces cancer. The best way to reduce TOR signaling is to avoid high protein. What's high? Initially, I said above 1 gram per kilo per day, which is what Joe mentioned. Then he also mentioned about 0.7, and I would say you could even go down to 0.6 if you really are sick. The protein content in breast milk is only about 1 gram per 100 ml. So daily protein intake is about 1 gram per kilo per day. You will never need more than that. I don't care how much you build muscles. You won't be able to use more than that. This is a study I published with Duke University that showed that a high-fat, very low-carbohydrate, very moderate protein diet basically had the same effects on hormones and other metabolic factors as severe calorie restriction. I got 30 seconds. Your health and lifespan will be determined by the proportion of fat versus sugar you burn over a lifetime, and that will be determined by the communication of insulin, IGF, leptin, and especially TOR. Cancer is not glucose-driven, nor is it driven by mitochondrial dysfunction. It is driven by growth signals, the most important of these being mTOR. Now, the good part here I should mention is the diet will be essentially the same. There are some tweaks we can make to it if one understands this. One understands the importance of protein, and there are certain amino acids that actually stimulate TOR more than others. It's TOR you have to keep down. Fortunately, when you're going on a so-called ketogenic diet, I hate the name ketogenic diet. I think Joe mentioned that. It should be a high-fat diet. It isn't ketogenesis that you want. It's a nice side effect. You want to burn fat. Life is not in the parts. We are all made of the same stuff. It's what you do with the parts that determines your life and your health, and that is going to be largely determined by mTOR, as it makes the decision whether cells should replicate or allocate resources towards repair. There we go.